Besides the first lines of defense and various protein systems that are activated in cascades in the blood and body fluids, several other cells play a crucial role in innate immunity. Generally speaking, cells of innate immunity can be classified according to their bone marrow origin into myeloid or lymphoid. Most of the myeloid cells, which will soon be described in detail, act as phagocytes. That is, they come into contact with pathogens, recognize them via specific receptors, and may be more or less effective, depending on the cell type, in phagocytosing and destroying them, thereby setting a series of responses in motion which subsequently influence other cells, both immune and not. Let's take a closer look at these cells and how they differ in their specific functions. Macrophages are certainly among the most important phagocytes in the body. They can derive either from progenitors in the tissues or from monocytes in the blood, which can differentiate into macrophages after leaving the bloodstream, driven by MCSF, macrophage colony stimulating factor. They are particularly abundant in connective tissue, submucosa of the gastrointestinal or respiratory tract. Moreover, they are given specific names in certain areas of the body. In nervous tissue, they are referred to as microglia cells, while in the liver, they are known as Kupffer cells. Both microglia and Kupffer cells populations can be self-sustaining thanks to the IL-34, which acts as the same receptor as MCSF. Besides their potential to become macrophages in tissues, monocytes perform specific functions that are important for innate immunity. There are two populations of monocytes in the blood. Classical monocytes, which make up 90% of the population and express CD14, a co-receptor of TLR4, and MD2, two pathogen receptors. Their role is to extravasate into tissues and, in case there are pathogens, to activate or differentiate into macrophages to promote inflammation and damage removal. Patrolling or surveillance monocytes, which make up the other 10% and express CD14 and CD16, which is the receptor for the Fc fragment of immunoglobulins. They mostly remain attached to endothelial cells, do not go into the tissues, and do not differentiate into macrophages. They should be responsible for watching for possible damage to the endothelium. Alongside monocytes and macrophages, granulocytes are another important class of phagocytes, and neutrophils play a key role among them. Unlike macrophages, neutrophils are not present in tissues under physiological conditions, but are only recruited when needed, infection or damage. They have the highest phagocytic activity, but live for a short time, unlike macrophages. They are therefore crucial in providing a first-line response. There are also dendritic cells which reside in the lymph nodes and peripheral tissues. There are two types of dendritic cells: conventional dendritic cells, CDCs, and plasmacytoid dendritic cells, PDCs. Only CDCs are myeloid elements, while PDCs are a less represented category and have a lymphoid origin. Unlike the other myeloid cells, CDs are not involved in first line of defense, but more in processing antigens of phagocytized pathogens and representing the antigens to the lymphocytes of adaptive immunity. But how does phagocytosis occur? Whichever phagocyte is involved, the process of phagocytosis takes place through common steps. Phagocytes can come into contact with pathogens thanks to the presence of certain phagocytosis receptors. As a result of this binding, the phagocyte is able to progressively absorb the pathogen in a vesicle that is called the phagosome. The phagosome then comes into contact with lysosomes, which merge with it and form the phagolysosome, a structure which is fundamental for the destruction of the pathogen and the release of antigens that will then be exposed on the cell surface to stimulate adaptive immunity. Pathogen killing occurs by various mechanisms. 1. Lowering the pF in the phagolysosome due to the acidity of the lysosome. 2. Presence of specific antimicrobial enzymes or peptides, especially in the primary and secondary granules of the neutrophils, which can fuse with the phagolysosome. 3. Synthesis of reactive oxygen species ROS and nitrogen species RNS, done by specific enzymes. Finally, it is worth noting that phagocytosis is not the only way a cell can take material from the outside. There are other mechanisms, such as receptor-mediated endocytosis, which also occurs in other cells than phagocytes, and macropinocytosis, which consists of ingestion of large quantities of extracellular fluid. The latter is particularly important for immune purposes for dendritic cells and macrophages, as it enables them to perform a proper immunological filter action and detect pathogen components in body fluids at an early stage. But how do phagocytes recognize pathogens and phagocytose them? What are the receptors involved in this process? We distinguish three groups of phagocytosis receptors. 1. C-type receptors of the lectin-like family, so called because they have one or more CTLDs, a C-type lectin-like domains, containing a CRD carbohydrate recognition domain. Scavenger receptors, complement receptors, and immunoglobulins Fc fragment receptors. The first group includes dectin-1 and the mannose receptor. Dectin-1 is structurally characterized by a single CTLD and is strongly expressed by macrophages. It binds glucose polymers with beta 1.3 type binding typical of fungi walls. The mannose receptor, on the other hand, has numerous CTLDs and is expressed not only by macrophages but also by DCs. It performs a recognition function towards different mannosylated ligands typical of various pathogens, viruses, fungi, bacteria, etc. Scavenger receptors, on the other hand, are a broad class of receptors that owe their name, scavenger means sweeper, precisely to the ability to remove a wide range of molecules and maintain the body's homeostasis. These include amniotic polymers, lipoproteins, apoptotic cells, esterified cholesterol, phospholipids, etc. There are several classes of scavenger receptors, but two of them are the most specialized, Class A and B. Class A scavenger receptors are structurally characterized by trimering of collagen domains capable of binding various components of the bacterial wall and facilitating internalization. SRA1, SRA2, and MARCO, macrophage receptor with a collagen structure, belong to this group. Class B scavenger receptors, on the other hand, do not have a collagen structure and share the ability to recognize LDL or HDL lipoproteins. This group includes CD36 and SRB1, two receptors that are expressed by macrophages and play a pathogenetic role in the development of atherosclerosis, a disease characterized by the accumulation of lipids in arterial walls. In conclusion, myeloid cells play a fundamental role in innate immunity. Most of them are capable of recognizing pathogens, internalizing and eliminating them. At the same time, this recognition activates a series of responses aimed at establishing an inflammatory environment to help eliminate the infection.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.